We're going to talk about cell structure. That's very important for your biology study. So let's first introduce like why cell structure and function is so important. Because because cell structure and function are primarily determined by 4 factors: matter, energy, organization, and information. So basically, in this chapter, we're going to talk about all kind of like organelle within the cell. What's it made of? What kind of like relationship between the different organelle? That's organization. And then within each organelle, what kind of a chemical inside? Why this? How does each organelle gonna work? And what kind of interaction between each other? And what kind of information it can give us? And then, so first, let's talk about prokaryotic cells, because prokaryotic cells is like, what is a prokaryotic cell? Prokaryotic cell is always very simple cell, like bacteria, virus, this kind of microorganism. They are small. They are relative to like like for example, plants, animal, and cells. They are much easier. So first, let's talk about the division, category of cell type. So basically, all form of life can be divided into 2 categories, which is prokaryotic and eukaryotic. We will first talk about the prokaryotic because it's simple. Prokaryotic cells, it doesn't have a membrane enclosed nucleus. So which means a cell outside is a cell membrane, and then inside there's like a nucleus. This nucleus is very important for each cell, because nucleus is the place where the DNA genetic information was stored. But so like for example, whether it's in prokaryotic cell or eukaryotic cell, so all the DNA genetic information are stored in the nucleus. But the difference between prokaryotic cells and the eukaryotic cells is just in eukaryotic cells, the nucleus, there's a membrane outside. But for prokaryotic cells, like nucleus doesn't have the membrane. So from an evolutionary perspective, the 2 categories of organisms that are composed of eukaryotic cell are bacteria, are Qian. So basically, this means prokaryotic cells can divide into bacteria and archaea. Both of these types are the microorganisms, are very small. With the cell size, it's between 1 micron and 10 micron. Bacteria is everywhere. It's around us. Like in the winter, a lot of people get sick because they get affected by bacteria. We can also find bacteria in soil, water, and like for example, when we are talking about bacteria, sometimes it's not only bad. Some like bacteria which cause the disease, they are really bad. But like in our digestive tract, like for example, in our stomach, there's a lot of like beneficial, like good bacteria. So a lot of like scientists, they do research. They find out like if we don't have those kind of good bacteria in our stomach, we'll get sick. Like a lot of like, for example, if you trying to get rid of, if you take some kind of probiotic, like for example, if you get sick, you eat some kind of antibiotic drug. And those kind of antibiotic drugs, sometimes they are not like target orientated. So which means they not only kill one type of specific bacteria, they kill a lot of bacteria inside. Sometimes they kill like good bacteria. Then you will get sick. Then you need to take some like prebiotic cells to add in those kind of good bacteria. So that's one good things I need to talk about. So that's why this book said most bacterial species are not harmful to human beings, and they play a very important role into ecology. However, some species are pathogenic, which means they are toxic. They can cause disease. Example of a pathogenic bacteria including Vibrio chiloria and the other type can cause anthrax. Like another type of prokaryotic cell is called archaea. Archaea is always found throughout the world. Also, they are less common than bacteria and always occupy extreme environments such as hot springs and deep sea winds. So let me Google online. So give you some example about archaea. Bacteria. So like, for example, let's read about the Wikipedia. You can see my website, right? So
Yes.
archaea, it's a domain of a single-celled organism. The microorganism lack cell nucleus. So this type of like archaea, they don't even have nucleus. And then so archaea are a major part of Earth's life. They are part of a microbial organism. In the human microbiome, they are very important in gut, mouth, or the skin. So besides bacteria existing in human, there's other like very good archaea existing in our body. So let's see the difference between. You can Google the difference between these two. So you can see archaea and bacteria have several different, including their cell walls, cell membranes, and the ability to survive through extreme conditions. Like for example, archaea cells walls are made of different polysaccharide protein and don't contain peptidoglycan, which is found in the most bacterial cell walls. So like, for example, the cell wall, the component of cell wall for archaea and bacteria are different. Like bacteria, they have like cell membranes are made of a peptide glycan. And then, but archaea cell wall made of like polysaccharide and the proteins. And then cell membrane, so like cell membrane, archaea and bacteria, they have different lipid composition in their cell membranes. So cell membrane, their lipid composition is different. First is cell wall and then cell membrane. And then archaea can survive in extreme condition, like very hot, temperature very, very high. But like a bacteria cannot survive in this kind of extreme environment. Like most times bacteria will existing in normal condition, like where the human being can survive. They cannot live in like very cold, very hot place. So that's a different three things: cell wall component, cell membrane component, and how like surviving environments. That's a different. Let's go back. So first, then this is so this chart give the structure about the bacteria cells. So inside, this is a nucleate. So this is like the red like thread. These are DNA. So it doesn't have a membrane, right? I'm talking about the bacteria. Bacteria doesn't have like a membrane outside of the nucleus. And then This is a plasma membrane. Okay, so the whole thing is a cell. Okay, this is the let's start from outside. Like for example, this is a pili. This kind of thing is like a pili. It's like a hair thing. Allow bacteria to attach to the surface or to each other. So like for example, when you got infected into by the bacteria, always those kind of organelle. It's like how to say it? It's like something grab on it. So like for example, our throat get some kind of affected. Those kind of bacteria will use this pili to grab onto our throat skin, and then you will attach there. And then this is called a flagellum. Like this kind of flagellum is like a feet. It's like a feet for bacteria. They can move freely. And then next is like the most outside. It's called a glycocalyx. It's a covering. Like you from the name you can know it's component. It's made by glycan, which carbohydrate. It's called glycocalyx. And then next one inner membrane called cell wall, provide support and protection. So this is more like give the structure of bacterial cell. And then the next level is plasma membrane. Okay, so this plasma membrane will separate the organelle of bacteria inside with the outside environment. And then this is the nucleate. DNA was existing. And then these small things is called ribosome. Ribosome is the place where protein was synthesized. This is cytoplasma, site of the metabolism. So this is like a swimming pool. This is like water, like swimming pools. And then when like inside of a cell, there's a lot of chemistry going on. Like for example, in order for protein synthesis, like beside the ribosome have to work, there's be a lot of other chemical reaction going on. Like for example, transcription, RNA involved. They all happen in the cytoplasma. So this picture, so this is like you cut it open, and then this is like TM. TM is it's one type of like electronic microscope. It can see this E. coli is a bacteria. How it look like? It's like this. So outside is a filling. This is like flagella, and then this is like this shape. So everything here just inside of here. So okay, I think I talk about everything. So one thing what I should mention about it is that nearly all species of bacteria and are killed. So almost, so it means all type of prokaryotic cell, they have a rigid cell wall. So this is rigid cell wall. Why is rigid? Because the purpose of the cell wall is to support and protect the plasma membrane and the cytoplasma. So these have a rigid. It have to protect the rest inner structure. So the cell wall composition varies widely among prokaryotic cells, but commonly contain peptides and carbohydrate. So cell wall most of the times contain peptide and carbohydrate. Peptide we mentioned about, right? It's a small fragment of a protein. Carbohydrate we talk about is a glycan. So it's also very porous, allowing most the nutrient in the environment to reach the plasma membrane. So there's be a lot of like hole outside of on the surface of a bacteria. So the bacteria can get a nutrient in and out. So many bacteria also secrete glycocalyx outer viscous covering surround the bacteria. The glycocalyx trap the water and helps protect the bacteria from dying out. Certain strain of bacteria that invade animals' body produce a very thick gelatinous glycocalyx called the capsule that may help them avoid being destroyed by an animal's immune system or aid as attached to the cell surface. So this is glycocalyx. It's very important, not only and keep the structure and the protection from outside. It can also help the bacteria get destroyed by outside environment, and even can help the bacteria to attach to the cell surface. And then that's all. Then we are talking about the eukaryotic cell. They are much more complicated than prokaryotic cells. So this type of cells include protists, fungi, plants, and animal. So parasite, paramecium, and algae are types of protist. Algae is like some kind of sea plants in the ocean. This is called a protist. I mean, they're not like plant like tree cells or like leaf cells, that's plant, but algae, it's kind of different from plant cell. Structurally, from the structure, they are different things. They belong to protist, and the yeast and the mold are type of fungi. Their structure is different from plant cell, animal cell, and the protist. So basically, when the scientists divide the eukaryotic cells into the 4 divisions, it all depends on the difference of their structures. And then animal cell is different. Animal cell is different from the rest 3. But there's something in common related to all the 4 category. So first of all the eukaryotic cells possess a true nucleus, where most of the DNA is housed. So like, for example, this is bacteria. It's very simple. So even though it's not like a real nucleus, it's just like they don't have a membrane. It just DNA was accumulated here. But here, see, this is a nucleus, right? It's more complicated than a prokaryotic cell nucleus. So the whole purpose thing is, so this nucleus, they have a membrane, right? And between on the outside, like that's all the hole. This hole called the nuclear pole. It can through these holes, molecule can pass in and out of the nucleus, and then this is red thing still be like DNA. And then beside like this is DNA, there's other things called nuclears. Okay, so basically this is the ribosome subunit assembled here. So basically the protein, this is a nucleus. So compared to the bacteria, this ribosome is here, right? It's outside of the nucleus. But in the eukaryotic cell, this is animal cell. This is animal. You cannot say this is just for animal. So this is animal. So for animal cells, the ribosome also located inside of a nucleus. So basically, in animal cells, the protein was produced within the nucleus. So compared to the prokaryotic cell, eukaryotic cell exhibit extensive compartmentalization, which means they have many membrane-bound organelles that separate the cell into different regions. So like, for example, this is the one example of animal cell. So you can see there's a lot of like separate thing here. You can see like nucleus, they have a membrane. It's individual. It's separated with the rest. And then, for example, this is the mitochondria. It has a membrane. It's individual, separate from the rest. This is like Golgi. This is like cytosol. they all have their membranes. They're like individual compartment here, not like bacterial cells. Everything just merged together. So this kind of cellular compartmentalization allows a cell to carry out the specialized chemical reaction in different places. So which means in prokaryotic cells, because prokaryotic cells, they're very simple organisms, which means there's still a lot of chemistry going on within the cells, but the number of reaction was very limited, and then all these kind of reaction most times is just happened in one place, like just like cytoplasma. But since animal cells, they are all more like at one cell type, there's much more chemistry going on within the cells. So, but even though there are tons of like much more chemistry going on compared to prokaryotic cells, but they separate everywhere. Like for example, if you want to produce the protein, it's within the nucleus. So like Golgi is used to produce the lipid and modify protein. So this type of reaction will only happen in Golgi, right? They separate from the rest of the function of the cells. So we can, let's look at this picture. It's very important. So we're gonna learn a lot from here. So it's better you spend some time, look at the picture and try to remember as much as possible about the organelle within the cells and understand their function. Let's do one by one. Like for example, this is the big purple thing. It's called the nucleus. It's nucleus. Okay, try to differentiate between this nucleus and the nucleus. Okay, it has additional O between. When you talk about nucleus, it's a whole thing. It's outside. So this big thing, the whole purple thing called nucleus, is the area where most of the genetic material is organized and then expressed. And then this nucleus is just this is where ribosome were assembled. Okay, so protein synthesis will happen in this blue ball. And then the red thing, it's called a chromatin. Chromatin is like complex of protein and the DNA. So this is envelope, double membrane that encodes the nucleus. See, this is nucleotope, which means the nucleus membrane outside to protect and separate the nucleus with the rest organelles. And I just mentioned about nuclear poles. And then this small one, is called a lysosome. It's a site where micromolecules are degraded. Like for example, when there's some like a protein, they have no use anymore. The cell need to get rid of it. So the cell will send the unused protein or other like lipid, big molecule will send to here. It will have an enzyme help to degrade all these kind of micro molecule to make it from big molecule to small molecule, and then completely get rid of it, and then be transmit to outside of the cell. This is ribosome. So, and then this kind of yellow thing, plasma, this is membrane, cell membrane. It's different from nucleus membrane. And then, okay, this is cytosol. So it's like a cytosol. It's a little bit similar to cytoplasma in a bacteria. So many many reactions were happening here. This is Golgi. Golgi is cytomodification and sorting of a sequence of lipid and protein. So like for example, protein was made here, and then but the protein you synthesize within the nucleus. Nucleus is not like it cannot function very well, and then it's needed the protein inside of the nucleus need to transport outside, and then to do some modification here. Modification include a lot of add some like carbohydrate on the protein, or add some kind of lipid on the protein, and then only those modified protein which happen in Golgi can be useful, can be functionable. And then this is peroxisome. It's the hydrogen peroxide and other harmful molecule was broken down. This is more like small molecule place. Like if some kind of small molecule or toxic small molecule, it's useful and it's toxic to cells, then the cells will deliver those kind of harmful small molecule to get rid of it. This cytoskeleton is like a bone. It's a protein filament that provides shape and aid to movement. So it's help the cell have its structure, and it's like a bone of a human body. It's keep the structure of the cells inside mitochondria. This is mitochondria. It is site of ATP synthesis. Like for example, when like for example, our human being, we have to live like for example, that's very important cellular chemistry going on. They're called cell respiration. Why cell need a respiration? Cell respiration, so basically convert the carbohydrate into the water and the carbon dioxide. And the most important thing is produce ATP. ATP is energy resource for the organism. So only cell can do respiration and then can generate ATP for usage. This ATP and cell respiration most times happen in mitochondria. We can talk about those kind of cell respiration later on, but like cell respiration, there are several steps, but the most important and steps will happen in mitochondria. I'll talk about the details later on. And then this is like smooth ER. That's a side of detoxification and lipid synthesis. So this is another place for lipid synthesis, and also it can get rid of like some toxic substance. And this is smooth. Why it's smooth? Because this is rough ER. Why I'm saying this is smooth, this is rough, because the rough ones, they have this kind of dot, purple dot outside, like something like there's some kind of dot outside, it will make it more rougher. So this one with rough with like spot outside, it's called rough ER. But this one, it didn't have any like spot outside. It's called smooth ER. And this one is another organelle, central centrosome, site where microtubes grow and centrioles are found. So this kind of microtubes and the centrioles, it's like a structure. It's like a bone in a human body. It's help to maintain a cell structure. So try spend some time to look at this picture and try to remember all the organelles and its function. It's very important. So let's talk about, okay. See, we just talk about like this is animal cells. Then we can talk about some plant cells. Plant cell and the animal cells, they have some similarity, but plant function different from the animals. So there's some difference too. Let's do one by one. So both plant cell and animal, they have a nucleus, right? It's purple. And then they all have like nucleus for ribosome subunit assemble. And then they have like chromosome. They also have a nuclear envelope, nuclear pore. Okay, and the ribosome too. They also have a smooth ER and a rough ER. They also have plasma membrane. But one big difference between the plant cell and the animal cell is the cell wall. So animal cell, they only have a plasma membrane. They don't have a cell wall. But only plant cell, they have a cell wall. It's additional wall outside of a plasma membrane, can provide like cell support. Okay, they also have a peroxisome. right? They have a Golgi, they have a cytoskeleton, and another big difference is called chloroplast, right? It's because animal, they don't have to do photosynthesis, but plants will need to do photosynthesis. What is photosynthesis? So photosynthesis basically is convert energy from the sun into the carbohydrate, right? So basically those carbohydrate were stored as energy within the plant cells, and then animal eat the plant. They can have they can get a resource of carbohydrate from the plant, right? So plant need to have those kind of capability to make the carbohydrate, right? This is chloroplast. It's the additional organelle for the plants can get in the carbohydrate from the sun energy. This is the mitochondria. It's the same thing. And then cytosol, the same thing. Another big difference is this central vacuole site that provides storage and regulation of the cell volume. So basically you can regulate the cell's volume by this kind of organelle. So another thing, let's talk about here. So we already talked about the plant cell and the animal cell. So most of the organelles are surrounded by a single or double membranes. So that's another thing I need to mention about. Research has discovered the cell can also become compartmentalized by a second mechanism called the liquid phase separation, in which aggregate solute such as protein and RNA molecules separate from bulk solvent and form a droplet. This droplet has a sphere shape with a measurable surface tension and viscosity. Molecules can diffuse within a droplet and occasionally leave it to pass into the surrounding liquid phase. Then another thing. So that's one thing. The character of a cell are largely determined by the protein it makes. So many organisms, such as animals and plants, are multicellular, meaning that a single organism is composed of many cells. But the cell of most multicellular organisms are not all identical. For example, your body contains skin cells, neuron, muscle, and many other types. So that's a different between a different cell. Like for example, certain protein found in skin cells may not be produced in neuron and virus. Like multicellular organisms, they have different type of cell, and then each cell they have different structure. They function a different thing, right? This is a good example. Skin cell and neuron may produce the same protein but in different amounts, right? That's all. Amino acid sequence of particular proteins can vary in skin cells and neurons. Skin cells and neurons may order their different protein different ways. So, which means in multicellular organisms, different cells are different. They are different shape, different structure, and then they function differently. And surface area and volume are critical parameters that affect the cell size and the shape, right? So most of the cells, they are in very small size. For example, most bacterial size about 1 to 10 micron, and the eukaryotic cell is a little bit larger, 10 to 100 micron. right? So size is different for different type of cell. Why are cells usually so small? One key factor is the interface between a cell and its extracellular environment. That is, cell's plasma membrane. For cell to survive, they must import substance across their plasma membrane and export waste fruit. A cell within a large internal volume will require a great amount of nutrient uptake and waste export. So like, for example, those kind of import and export requires energy for the cells. So that's why cells always keep in a small size, so they can save the energy, save the space for import and export. And then the rate of the transport of substance across the membrane of a given cell is limited by its surface area. Right. So this is like a different size of the cell it's talking about. So a critical issue for sustaining a cell is the surface area to volume ratios, right? So as the cells getting larger, the surface areas of the plasma membrane increased with the square of the radius. So like, you can say this is the area. It's equal to 4 pi r squared. So if the r is like radius of the cell, when it's getting bigger, the surface area is getting bigger, and the volume also getting bigger. So, but when they divide together, and then r would be down. So that's why L radius cell getting larger. The surface area divided by volume will getting smaller. Right. So it's better to keep the cell into the small size, because a high surface to volume ratio is required for cells to sustain adequate level of nutrient uptake and waste. Okay, then we're gonna talk about each single one. Let's talk about each single organelle in details. First, we're gonna talk about the cytosol. What is cytosol? Cytosol is this kind of yellow stuff here. The region of a eukaryotic cell that is outside of the organelle, but inside the plasma membrane is this one, right? This like with the color is the organelle. This cytosol is outside of the organelle, but this is the plasma membrane, and then it's inside of the plasma membrane. Okay. So why this cytosol is important? Because synthesis and the breakdown of molecule always happened in this kind of yellow region. So first, let's introduce a concept called metabolism. So you must have heard it before. Metabolism is defined as a sum of the chemical reactions by which cell produce the material and use the energy necessary to sustain a life. So even though many steps of metabolism also occurred in the cell organism, the cytosol is the central coordinating region for many metabiotic activity of eukaryotic cells. So which means all those kind of chemical reaction, where did it happen? Those kind of metabiotic reaction, it's all happening in this yellow region, cytosol. So this kind of a metabiotic pathway is complicated. In each step of the metabiotic pathway, it's catalyzed by a specific enzyme. So what is enzyme? I mentioned about this before. It's a protein that accelerates the rate of chemical reaction. We're gonna talk about more, like you're gonna hear a lot of like enzyme later on. And then so some metabolic pathway involve the breakdown of a molecule into smaller component. This kind of process turned catabolism. So which means catabolism is just break down something, right? Such pathways are needed by the cell to utilize energy and also to generate a molecule that provide the building blocks to construct a micromolecule. So compared to the catabolism, so which is break down the big stuff into the small stuff. There's another metabolic pathway is called anabolism. So anabolism is the reverse way. It's a synthesis of the molecule and the micromolecule. For example, polysaccharides are made by linking sugar molecule. To make a protein, amino acid are converted and connect to form a polypeptide using the information within the with the mRNA. So like for example, making the polysaccharide and making the polypeptide, it's all like that belong to the anabolism because it's making stuff, right? And okay, now we're gonna talk about cytoskeleton. Cytoskeleton is like these kind of things. It's like a bone to human. It provides cell shape, organization, and movement. So cytoskeleton is a network of 3 different type of protein filaments. So cytoskeleton can be divided into 3 types: microtubes, intermediate filament, and the actin filaments. Each type is constructed from many protein monomers. So this is a big organelle, but it's made of the many different type of protein. The cytoskeleton is found primarily in the cytosol, so it's always found within the cytosol. Okay, and also in nucleus, along the inner, it can also find in the nucleus. Let's talk about the cytoskeleton structure and their role in the construction and organization of cells. Right. So basically, the cytoskeleton can be divided into 3 types. Let's talk about one by one. Microtube. It's about 25 nanometer, and it's a hollow tube thing. It's like this. So this is a good example, like this. It's made of the protein, hollow tube composed of the protein tubulin, right? All these kind of microtubes are made by the protein called tubulin. And then its function is help the cell to keep its shape. It can help the cell to organize the cell organelle. It can also chromosome sorting in the cell division. It can function during the cell division. It can help the component can move within the cells. It's also the big component of like organelle cilia and the flagella. So it's the main component of this type of the organelle, which can help a cell move. And then another type is called the intermediate filament. It's about 10 nanometer. Its structure is a twisted filament. Okay, like this. It's called a twisted filament. It's composed of different protein, including keratin, laminin, and others that form twisted filament. Okay, it can help cell shape, provide the cell with mechanical strength, anchorage of the cell and the nucleus membrane. The last type is called actin filament. It's about 7 nanometer. And the structure is like a spiral filament, like this kind of shape. It's almost made of like protein, like two intertwisted strands composed of the protein actin. So it's made of by protein actin. It can help like cell shape, strength, muscle construction, like intracellular movement, cargo cell movement, cytokinase in animal cell. Okay. So I just mentioned about these 3 types. And then, so some proteins, like motor protein, will interact with this cytoskeleton to promote movement. So what is motor protein? Motor protein are a category of a protein that's use ATP as a source of energy to promote various type of a movement. So basically, this motor protein can interact with the cytoskeleton, and then by this kind of binding, the cytoskeleton can get energy from the motor protein, and then can move within the cell. That's how it works. So this is structure of the motor protein, and then it will, and then this is a picture how motor protein will interact with the cytoskeleton, right? This red one is cytoskeleton. It will attach to the cytoskeleton by this way. And then how does it work? So this picture shows how the motor protein move along a cytoskeleton filament. So when it's attached, first it will release, like detach from cytoskeleton. And then head cocks forward and bind to the filament. So first it's attached to this part, right? This part, and then it's get off, and then it's from this shape become the other shape, right? That's called head cocks forward and bind to. So it will twist a little bit, and then from here to become here. It's attached to this one, right? Then it moves, and then this is back forward, and then this kind of back forward can help the tail move forward. That's how it this motor protein interact with the cytoskeleton, and how it's like the way how it moves. So motor protein can cause 3 kind of a movement. Movement of cargo through via the motor protein, movement of the filament, and the binding of the filament. So this is why this is like motor protein. It's move along the cytoskeleton. It can, first of all, it can carry the cargo, right? When it moves, it carry the cargo, and then with the movement of cargo, it can transport the cargo to somewhere. So basically the cargo will attach to this tail, right? Before the cargo was here, and then with the movement, it's transferred to here. So it's help to carry the cargos to somewhere. And then there's another function called movement of filament, like a motor protein called myosome can retain in place and cause the filament of a skeleton to move. So like, for example, this is a good example about motor protein move from here to here. And then this is purple thing is a cargo. So it will bring the cargo from here to here. This is called the mitoprotein moves. There's another thing called filament moves. So basically, this time, this motor protein, they didn't move. They just stay somewhere. This flag, like cytoskeleton filament, they will move, right? Before it's here. Now it's moving forward. But the motor protein was kept in the same place. This is called another way called filament band, right? This is the motor protein. And the lingular protein. Both the motor protein and the filament are fixed in place. So the action of the motor protein cause the microtubes to bend before it's here. Like this is the microtubes, and then motor protein will be attached in between. And this kind of like motor protein, it will change the shape. Because of the change shape, it's also make the microtube bend. That's called bending of a filament. This is three type of like interaction between the motor protein interaction with the cytoskeleton. Then also it's can transport the cargo. It can make the fragment like a cytoskeleton moving and helping it bending. That's how it works. Then we're gonna talk about another organelle. It's called the cilia and flagella. Because this is like this two organelle allow cell to move. Okay, so the difference between this cilia and the flagella, it's flagella is longer than cilia, and are typically found singly or in pairs. So, like for example, this is a good example. This is sperm, right? Sperm head, and then at the end, there's some kind of flagella, right? Like a tail. This kind of flagella is tail. It's like it can swim, right? Change the shape, helping the sperm move. And then this is another type of cell outside that's long to flagella, and those kind of flagella, it moves, help the cell move. And then this is another type of cell. It has a lot of cilia outside. Those kind of small cilia that it's like help the cell swim in the environment, help it move. So both the flagella and the cilia cause movement by generating the bands. Let's move along their length and push backward against the surrounding fluid. Right? Like for example, I mentioned about this. Okay, and let's talk about the structure. Even though they are different in the length, but these two flagella and the cilia, they all have the same internal structure called axoneme. So this is structure of axoneme. So like for example, this is like a big picture of the cilia. This is the structure. And then when you trying to cut the slide and make it bigger, you can see they are composed of a basic unit called the axoneme. The axoneme contains microtubes. Right. Okay. For example, in the center, in the center. This is called central microtube pair. Central microtube pair. That's like, here is like triplet microtube. We have 3 together. This is like 2 together. And then this thing we call the radial spoke. And then in between the microtube, there's a protein, linking protein together. What else? This is like a dining arm, right? That's all its composition. And then in the cilium and the flagella of most eukaryotic organisms, the microtubes form the arrangement called 9+2 array. 1, 2, 3, 4. See, that's 9 here. 1, 2, 3, 4, 5, 6, 7, 8, 9, right? 9+2 in the middle. The outline are doublet micro, right? This is 9. Everyone is doublet microtubes, which is composed of a partial microtubes attached to a complete microtube. So this is complete microtube, and then this is part of. Each of these 2 central microtube are single microtube. Radial spoke project from the outer doublet microtube towards the central ones. The microtubes in flagella and cilium emanate from the basal body, which are anchored to the cytoplasmic side of the plasma membrane, right? And then at the basal body, the microtube form the triplet structure, much like the centrosome of animal cells. The basal bodies provide the site for microtube growth. That's the structure of the cilium. So there's more talking about the movement of both flagella and cilium involves the propagation of a band, which begins at the base of the structure and proceed forward to the tips. The bending occurs because of dining is activated to work towards the minus end of the microtubes. So basically, what it's talking about, this is dining arm was activated and worked towards the minor end of the microtube. So basically, this is activated, so it will move along forward to the microtube. So then we can talk about nucleus. So okay, the membrane that enclose the nucleus are part of a large network of a membrane called endomembrane system. So basically, he's talking about this kind of a black membrane outside. That's a nucleus membrane. This is called the endomembrane system. So this system not only the nucleus envelope, there's a hole in between, which enclose the nucleus, right? But also the endoplasm, and when the membrane goes to okay, this endomembrane system not only about the nucleus envelope, also enclose the nucleus, and also okay, he's talking about endomembrane system. He's talking about everything within this membrane, right? It's include the nucleus and all kind of organelle outside. This is like ER, like smooth ER, rough ER, Golgi. Golgi is here, this one, right? And the lysosome is here, right? So everything inside. It's endomembrane system. So everything is enclosed by this big membrane. Okay, now we're going to talk about chromosome. Okay, first let's talk about nucleus. Nucleus, the outside. This membrane structure is called nucleus envelope, right? Because it's a nucleus membrane. And then this is the, and then inside, this is a chromosome. So like this is like big picture of the nucleus membrane. We have two layers, outer membrane and the inner membrane. This is like these two layers called the nuclear envelope. Right? And then through this kind of the nuclear pores, it's the where the inner and the outer nuclear membrane make contact with each other. The pores provide a pathway for the movement of molecule and the micromolecule into and out of the nucleus. Right. So through this kind of hole, material can transport inside and outside of the nucleus. So, and then inside of the nucleus is this kind of a red thread. Okay, this kind of red thread, it's a chromosome, right? And also has a lot of like, for example, this red thing is chromosome. And then this blue thing is called internal nuclear matrix, right? They're all, it's represent this part. When you like zooming in, you can see more details, red and blue here, right? So each chromosome, like a red thing, it's composed of genetic information, namely DNA, and the many types of protein that help to come part of the chromosome to fit inside of a nucleus. So this red thread is have two parts. One is protein, another one is DNA. They compact together to make the chromosome. So the complex formed between DNA and such protein is called chromatin. And then this kind of nuclear matrix, the blue thread, it consists of two parts. The nuclear laminar, which is composed of the intermediate filaments that line the inner nucleus membrane, and the internal nuclear matrix, which is connected to the laminar and fill the interior of the nucleus. So this is the picture. All right. The nuclear matrix, the blue one, it serves to organize the chromosome within the nucleus. Each chromosome is located in a distinguished chromosome territory, which is visible when the cell are exposed to dye and label specific type of chromosome. Okay, so this color all represent the chromosome. Right. Because they are dyed with different color. So the primary function of the nucleus are the protection, organization, replication, and the expression of the genetic material. This topic, then we're going to talk about this later. Another important function is the assembly of a ribosome subunit, cellular structure involved in the producing the polypeptide during the process of the translation. The assembly of a ribosome subunit occurred in the nucleus, which is the drop-dead organelle in the nucleus of non-dividing cells. So basically, he talk about this kind of a blue thing. Blue thing, it's assembly of a ribosome. So this kind of blue things is the where the place protein will be synthesized. Then we can talk about ribosome. Ribosomes are composed of the two subunit, one small and one large. Each subunit contains one or more RNA molecule and several types of protein. So basically, ribosomes are composed of the RNA and the protein. Okay, so so the RNA which within the ribosome are made in the nucleus, but the protein within the ribosome are produced in cytosol and then imported into the nucleus through the nuclear pores. So ribosome protein and RNA molecule then enter the nucleus and are assembled into the ribosome subunit. So basically, protein were made outside and then transport inside of the nucleus and then combine with the RNA to become a ribosome. And then ribosome will assemble together, become this big blue ribosome subunit. And then this big blue ribosome subunits will be moved through the nuclear pores into the cytosol. Okay, and then and the protein can be synthesized within the nucleus with in the side of the cytosol. And then we can talk about the endoplasmic, like this organelle. It's like lifting. Okay, we just talk about the nucleus. Now we're going to talk about the ER. ER. So endoplasmic reticulum. This is the endoplasmic reticulum is the place to sort some of the protein and carry out a lot of the like metabolic function. So basically, it's a network of membranes that forms flattened, fluid-filled tubers, right? So it's like this. It's flat. It has a lot of fluid inside. It's tubers. Okay. There are two different ER. It's called a smooth ER and the rough ER. See, those kind of like, tubes without the dot outside, it's called smooth because it's smooth. And then if like ER has this kind of like dot outside, it's called rough because it's rough. So rough ER. So this kind of dot is ribosome. So on the like outside surface of the rough ER is started with the ribosome. So this kind of dot is ribosome, giving it a bumpy appearance. Rough ER plays a key role in the sorting of the protein that's destined for the ER, Golgi, all kind of other environments. So protein are packaged into the membrane visco. So move from one location in the endoplasmic membrane to another. So basically, rough ER it's help protein move from one organelle into another one. So basically, rough ER is used to help protein to transportation within the cells from one organelle to another one. A second function of the rough ER is the insertion of certain newly made protein into the ER membrane. A third important function of rough ER is attachment carbohydrate to protein lipid. So basically, it's modified the protein already made. Like when protein come to the rough ER, it can like do some kind of chemistry to modify it a little bit. And then, for example, one example is attach some kind of carbohydrate into the protein. This process called glycosylation. Okay, smooth ER. Smooth ER is this type. They don't have the ribosome outside. So their function can divide into three: metabolism. So this smooth ER membrane provide a large surface area for enzyme, play important metabolic roles. So basically, a lot of like reaction will happen on this surface because they're smooth. Enzyme can work here. Like for example, in liver cells, enzyme in a smooth ER detoxify many potential harmful organic molecule, including some kind of drugs and ethanol. So, which means in the liver cells, enzyme can help get rid of some toxin within the smooth ER. And then, how does this work? This enzyme can convert hydrophobic toxic molecule into more hydrophilic molecule, which are easily excreted from the body. Alcohol consumption lead to a great amount of smooth ER in liver cell, which increase the rate of alcohol breakdown. So smooth ER of the liver cell also play roles in carbohydrate metabolism. The liver cell of animals store energy in the forms of glycogen, so which is a polymer of glucose. So like for example, those kind of glycogen granular, it's very close to smooth ER. When the chemical energy is needed, enzyme activated break down the glycogen to like glucose to this kind of compound, and then the enzyme in the smooth ER called another type of enzyme in the smooth ER can remove the phosphate group and the glucose export from the liver cell into the bloodstream. So basically, glycogen was located very close to the ER. So when enzyme, like when our organism need energies, so some kind of enzyme can degrade those kind of glycogen, but glycogen cannot directly be converted to the glucose. It's we're converted to kind of intermediate. So it needs another enzyme on the ERs to further convert that intermediate into a glucose, and then glucose can be transported into the bloodstream to work. So which means smooth ER can help our body to get glucose as an energy resource. And smooth ER can also can accumulate the calcium, iron. And then this smooth ER, they have calcium pump transport calcium into the ER lumen. The regulated release of calcium into the cytosol is involved in many important cellular processes, like including muscle contraction in animals. So basically, the smooth ER can help regulate the calcium iron in the body, and those kind of calcium is very important for like, for example, a lot of cellular process, for example, muscle contraction. So when we need to contract our muscles, so we need ER to help us to regulate, to release the calcium for us to move the muscles. And then the last function about the ER, it can help make the lipid and do some kind of modification. So how it works, enzymes in a smooth ER, it's critical in the synthesis and modified lipid. For example, a smooth ER, it's a primary site for synthesis of phospholipid, like one of very important lipid we just mentioned, which are the main lipid component of a eukaryotic cell membrane. So Another thing is enzyme in the smooth ER necessary for third modification of lipid crosstalk to produce steroid hormones such as estrogen and testosterone. So like ER is very important for the lipid synthesis and the modification. These lipids are very important for a lot of like living organism function. Okay, now we're going to talk about the Golgi. With the Golgi, Golgi is this small one, it's the blue part, right? What's the function of the Golgi? Golgi direct the processing, sorting, and secreting of cellular molecule. So look at its structure. It's very similar to the ER, right? It's like this, very flat. It consists of a stack of flattened membrane, with each flattened membrane enclosing a single compartment. So there are two models being proposed to explain how material moves through the Golgi apparatus. One is called the viscular transport model. So material are transported between the Golgi, right? Through the membrane visco, that's but from one compartment in the Golgi and fused with another compartment. So basically, all those kind of material were enclosed in some kind of type of membrane, and those kind of like enclosed membrane visco will move between, like Golgi is several layers. It will move. Those kind of visco will move from one layers to another layers. That's how it moves through the Golgi apparatus. Another model is like it's called cysternal maturation model. So which means visco from the ER fused to form the cystener at the cis phase. The cystener that was previous at the cis phase become the median cystener. This addition of the cystener move the other median cystener towards the trans phase. So it's a little bit complex. So it's more like one by one. It's like one by one visco, one by one. It's not easy. It's just you have to more convert. Is there a picture here? Let's see. But this is like just a proposal. So let's talk about more function of a Golgi. Like for example, enzyme in the Golgi can process modified certain protein and lipid. Like carbon, how is modified protein and lipid? Like for example, one good example is carbohydrate can be attached to the protein and the lipid in the ER. But this kind of the same thing could happen in the Golgi. Okay. A second type of process is proteolysis, where enzyme called protease make cut in the polypeptide. So like for example, if some kind of a protein or polypeptide go through the Golgi, the enzyme on the Golgi can cut it. Like good example is a hormone, insulin. It's first made as a large precursor molecule termed proinsulin. This is like for example, that's why this insulin, how it's made. It's first like precursor called a proinsulin. It's a much bigger molecule. It's made somewhere, and then when this precursor of the insulin is moved into the Golgi, and then this precursor is packed with the protease into the visco, and this kind of protease, it represents one type of enzyme. This will cut off a portion of a precursor to create a smaller insulin, right? So basically, the how insulin was made, it's made on the surface of a Golgi, and then some kind of enzyme called a protease will cut the precursor into the small ones. These small ones is a real insulin. It's a real insulin we're gonna use. And then also the Golgi can work as can function as a secretion. How it works? The Golgi pack different type of material into a secretory visco. So it will pack different material into some kind of visco. And now this kind of visco will fused with plasma membrane. It will like this, right? A lot of like material will be enclosed into the membrane to form the visco. And then this kind of visco will fused with plasma membrane. And then when it fused, it will release outside. Then those content within the visco will be released. And then protein destined for secretion are synthesized into the ER and traveled to the Golgi, and then are transported by visco to the plasma membrane. That's how it works. What else can you talk about? And then talk about the lysosome. So what's lysosome for? Lysosome are involved in the intracellular digestion of the micromolecule. So what's lysosome for? It's used to break down molecule and the micromolecule, right? Within the lysosome, there's a lot of like enzymes. It's called acid hydrolase. What's this enzyme? Its function? It's a hydrolytic enzymes that use a molecule or want to break down a covalent bond. So basically, for example, this is the molecule. It's transported into lysosome, and then it will react with water under this enzyme. And when this molecule will be break down into two molecules, right? It will break the covalent bond between R1 and R2. That's what happened in the lysosome. This type of reaction called hydrolysis, because it's need one molecule of water for breaking down the covalent bonds. So the inside of lysosome has a pH about 4.8. Right? So which means if the lysosome, so for example, this type of enzyme only work under the pH 4.8. But when this kind of like lysosome membrane was breaked, this type of enzyme released into the cytosol. But cytosol, the pH was very neutral. It's about 7.2. So this is neutral environment, won't work for the enzymes. So under this kind of circumstance, the enzyme within the lysosome, it doesn't work anymore, right? So which means even the lysosome membrane is broken, the enzyme released in the cytosol, but this enzyme won't work within the cytosol. So which means there's a lot of like enzyme, other type of enzyme, or molecule within the cytosol. So even though this type of enzyme released in the cytosol, nothing will happen, which is good, right? So which means the breaking down of a molecule can only happen in the lysosome, not happening in the cytosol. That's good. So this different type of enzyme can break down a lot different type of material: carbohydrate, protein, lipid, and nucleic acid. So one function of lysosome involves the digestion of substances that are taken up from the outside of the cell through a process called endocytosis. So which means the cell can bring the stuff from outside, and then the lysosome can help to get rid of, degrade those kind of stuff from outside environment. I'm going to talk about the vacuoles. Okay. Vacuoles only existing in the plant cell, fungal cell, and certain protists. So inside of a vacuoles, it's not empty. They have a lot of fluid, sometimes even solid substance. So this is the picture of the volume of vacuoles. There's a lot of stuff inside, right? It has a lot of fluid inside. So most of vacuoles are made from the fusion of many smaller membrane of the vesicle. And then vacuoles in animal cells tend to be smaller than those in plants and are more commonly used to temporarily store material or transport the substance. Such vacuoles are sometimes called the storage vacuole. So then we talk about the function of the vacuoles. It has a lot different function. They differ among cell type and even with the environmental condition. Like, give an example, mature plant cell usually have a large central, so this is plant cell, it have a very large central vacuole, occupied 80% of more of the cell volume. The central vacuoles served two important purposes. First, it stores a large amount of water, right? Because cells, like plants, they need a lot of water, right? Most times it means water will be stored, will be contained within the vacuoles. And then beside that, water, it also have enzymes, ions, right? Inside of the vacuoles. It also have protein and a pigment, right? It can also perform a space filling function. The central vacuoles produce some kind of pressure on the cell wall, right? So this is cell wall. Central vacuole is very big. It's close to the cell wall. It's produce some kind of pressures. If a plant becomes dehydrated, which means losing water, this pressure was lost. Like I just said, there's a lot of water within the central vacuoles. So when it's losing water, this central vacuoles will shrink. It will become smaller, so it won't produce those kind of pressure to the cell wall. So this type of pressure is important to maintain the structure of the plant cells, and the plant cells help to drive the expansion of cell walls, which is necessary for growth. Right? So the central vacuole help. It's very important for the plant cell, for its structure, for its expansion of the cell wall. So certain species of protists use vacuoles to maintain cell volume. Freshwater paramecium have smaller water field. Okay, so this is like one type of protist, right? So it has those type of the vacuoles called the contractile vacuoles. Expand as water enter the cell. So like for example, this is some kind of protist. Then if water go through the cell wall and then go inside, so those kind of water will go inside of this kind of vacuoles. So it will absorb the water and expand. So it's it's trying to observe the water more and more. When it's observed the water, it's getting bigger and bigger. And once they are big enough to reach certain size, the vacuoles fuse with plasma membrane. So it's getting bigger and bigger. It will like touch the plasma membrane. It will fuse with the plasma membrane, expelling their content to the so it will release the material inside to the outside of the cell. This mechanism is necessary to remove excess water that continues to enter the cell by diffusion across the plasma membrane, right? It's getting bigger and bigger. It's will attach to the plasma membrane, and then it will release the waters, water. It will release the water, which previously been observed. Another function of the vacuoles is degradation, right? Some protists, they will like engulf their food into large food, like this type of called food vacuoles, right? It will observe the new nutrient outside in the process called phagocytosis. Food vacuoles contain enzymes break down those kind of nutrient micromolecule in the food, right? And then help. So basically, this type of food vacuoles will help microorganism to digest the food, right? First by observe it, and then with the digest it with the enzyme inside.